Okay, so today we are going to start with development of male gametophyte. Okay, so in development of male gametophyte, first of all we need to study that what is male gametophyte, right? So quickly I will revise the content. Last time we have studied about flower. Flower is a sexual reproductive organ in plant. Okay, so at the center there is a gynoecium. This is gynoecium, right? And on the either side there are androecium like this. Okay, so these androeciums are called the male part of flower. Okay, now today we are going to study that in this androecium it is actually divided into two parts. Okay, this androecium, so if I draw this androecium here down, so this is the upper part of androecium, and this is the fine stalk of this androecium, right? Now this upper part is called as anther. This upper part is called as anther. Okay, now this anther is bilobed. It means it has two different partition, like our human heart has 4 chambers. Okay, and it is divided into 2 compartments. Similarly, this anther is also divided into 2 compartments, so it has 2 lobes, and each lobes contain 2 locules. Okay, each lobe contain 2 locules. Now in this anther, an important process by which the pollen grains are formed, that process we call it as microsporogenesis. Okay, so today we are going to study about microsporogenesis, and along with that we are going to study TS of anther, like what are the materials present inside the anther. Okay, so we'll take a cross sectional, like we'll divide it, if we cut it like this. So after cutting, so now when we cut this anther, okay, so you will see basically anther has 2 lobes, right? And in anther, pollen grains are formed. Now here we are going to study about how these pollen grains are formed in the anther. Okay, and when we are studying about development of pollen grain, we call it as microsporogenesis. Okay, we call it as microsporogenesis. Okay, now here students may get confused, like microspore, megaspore, anther. Okay, so I'll give you some homologies that you should remember. It will help you during your examination as well as when you are studying. Okay, homologies. For carpel, we are using the term megasporophyll. Okay, and for stamen, we are using the term microsporophyle, right? And for ovule, we are using the term Microsporangia, right? For embryo sac, we are using the term female gametophyte. Okay, and for egg we are using the term female gamete, right? Then for anther, okay, sometime there may be term like pollen sac also, so both are same. Okay, inside the anther we have pollen sac. So for anther or for pollen sac we may use a term Microsporangium. Okay, and inside this pollen sac pollen grains are produced. We call it as pollen sac, we call it as microspore. Okay, so these terms will help you to learn the process. So basically today we are studying microsporogenesis. So if you find your microspore, it is here. Okay, genesis means production. So whenever this term comes, genesis, like spermatogenesis, okay, or you can hear that gametogenesis. So genesis means production. Now here what is producing? Microspores are produced, right? So microspores means pollen grain, right? मैंने pollen sac लिख दिया, it's pollen grain. Okay, so pollen grains are produced. This process is called as microsporogenesis, right? Now let us first understand structure of Microsporangium. Microsporangium. So as just now we have studied, microsporangium means anther. So what we are studying? We are studying structure of anther, or you can say we are studying TS of anther. Okay, so structure of anther, or we are studying TS of anther, right? This is anther. If I cut this anther like this, okay, so after cutting it like this, the structure that you are going to get will be somewhat like this. Okay, after cutting, so in this there is two division. Here you can see, so this is the first lobe, as you can see here, lobe, and this one is the second lobe, first lobe and the second lobe. Now in this lobe you can see presence of two circular structures, one ear, second ear, third ear, and fourth ear. Okay, and this structure we call it as pollen sac. I'll write here, this is called as pollen sac, right? In this pollen sac, pollen grains are produced. In this pollen sac, pollen grains are produced. Okay, now see, when we are studying TS of anther, right? So anther shows two layers. One I can say it's outer one, and the second one I can say it is internal, outer and internal. Now the outer region is called as epidermis, and the second one, that is internal wall, this is actually called as wall of anther. Okay, this is actually called as wall of anther. And in this wall you can find two division. The first one is endothecium, and inner to endothecium you can find middle layer of cells. Okay, middle layer of cells. Right now this middle layer is actually made up of more than 2-3 layers. Okay, and together I can say that epidermis, endothecium and middle layer, all these layers together, they are giving protection for the development of pollen grains. Okay, now inner to this you can find presence of a layer, and that's called tapetum. Okay, that's called tapetum. Tapetum is, I can say, it's like innermost layer, and I'll write here it's an innermost, and it provides nourishment. Okay, it is innermost and it provides nourishment. Now since it provides nourishment, so it has dense cytoplasm because it is providing nutrition, right? And secondly, it may have more than one nucleus. Okay, it may have more than one nucleus. Okay, now let us see the structure. Okay, so it is somewhat like this, and as I said, here we have different layers, so you have to draw different layers. So first layer I'm drawing. So here you can see this is the outer one, that's called epidermis, and inner to epidermis we have internal layer, which is actually made up of many layers. So inner to that we have endothelium, and after that we have multiple layers over here. Okay, now immediately after this middle layer there is presence of pollen sac. Okay, so where it is present? Here. So these are called pollen sac, right? And in between, see now we need to understand where are the lobes. Okay, so if you draw the symmetry of this diagram, symmetry means half. Okay, so if I hide this one, so the remaining part is one lobe, and this is the second lobe, this is the second lobe, this is the first lobe. Each lobe have two locules, we can say, or two pollen sacs. So there are 4 pollen sac in one anther. Between this pollen sac you can find, between these two lobes you can find presence of one connective tissue over here. Okay, presence of connective tissue. So this is a connective tissue which keeps both the lobes connected to each other. Okay, now I was talking about this tapetum. So this is the tapetum, right? This is tapetum. So this is actually pollen sac, and on the surface of pollen sac you can find a thin layer, nutritive layer, which gives nutrition to the developing microspore, and this is called as tapetum. Here we can see tapetum. Okay, now tapetums are radially elongated and they provide nourishment to the developing microspore. Each anther lobe has 2 pollen sac, or we can say 2 microsporangia. Okay, 2 microsporangia. Now this pollen sac, this one, the pollen sac, I'll write here pollen sac. They encloses primary sporangious tissue inside them. Okay, and this primary sporangious tissue, they keeps on dividing mitotically to form sporogenous tissue. Okay, what does it mean? Development of pollen grain. Okay, development of pollen grain. Now see, anther, it has how many lobes? 2 lobes. Okay, so lobe 1 and lobe 2. In each lobe, how many sac? 2 sac. Sac 1, sac 2, and similarly here. Okay, now this sac, they are called as Microsporangium. Okay, these sacs are called as Microsporangium, and in this Microsporangium, like this is the Microsporangium. Okay, and these are the cells, with cells, tapetum. Okay, it's a tapetum, that is a nutritive layer. Okay, now in this nutritive layer you can find presence of these are called as primary sporogenous tissue. Okay, these are called as primary sporogenous tissue. Now this primary sporogenous tissue, they keeps on dividing, they keep on dividing, so I'll write divides and they redivides to form sporogenous tissue. Okay, to form sporogenous tissue. Now here I want to mention one point, like sporogenous tissues are formed. Okay, and this division is actually mitotic division. I'll write here mitotic. Okay, so when they are dividing mitotically, then the number of chromosome will remain same. Number of chromosome will remain same, that is they are diploid, right? Now this microsporogenous, now this sporogenous tissue, from down I'm continuing this side, this sporogenous tissue, they act as microspore mother cell. Okay, and they all undergo meiosis. Very important, huh? They are all undergo meiosis to form 4 haploid spores. Okay, so when meiosis term is used, we say haploid, and when mitotic word is used, we say they are diploid. Okay, so don't get confused in these terminologies. This is one of the microspore mother cell. Okay, so microspore mother cell, when it will divide, it will form 2 cells. So this is a division, 2 cells. Okay, and during meiosis process, total 4 cells are formed. So at last it will be producing how many cells? 4 cells, like this. This we also call as, because of 4 pollens, we say pollen tetrad. Okay, pollen tetrad. And later on these haploid spores ultimately they get separated. Okay, ultimately they get separated, and finally what is formed is called as pollen grain. Okay, that is called as pollen grain. Okay, so now we'll quickly revise what we have done. So first we have studied about anther. Okay, and where anther is present exactly I have shown you, and then after I have shown you that the anther is called as microsporangium. Okay, and in this microsporangium pollen grains are produced. This process of formation of pollen grains are called as microsporogenesis. Then we have studied certain homologies, like carpel is called as megasporophyll, stamen is called microsporophyll, ovule is called megasporangium, then embryo sac is called female gametophyte, egg is called female gamete, anther is microsporangium, pollen grains are microspore. Okay, and then after we have studied about the TS of anther. So when we divide anther transversely, then we can get 2 different compartments. Okay, so I can say that the TS of young anther shows outermost layer called as epidermis, and inner to that we have another layer, that is wall of anther. Right, now when we enlarge this structure, it looks like butterfly shape. Okay, and the wall of microsporangium, this is called microsporangium. The wall of microsporangium is composed of epidermis, then after epidermis we have endothecium and the middle layer of cells. Right, inner to middle layer of cells we have tapetum. The epidermis is the outermost layer of microsporangium, and the cells of epidermis are generally stretched and flattened. The next layer, the layer next to the epidermis, is endothecium, or we can say fibrous layer also. Anther is generally bilobed, and it's containing 2 longitudinal running chambers. Okay, and that chambers are called pollen sacs. Each chamber may contain, they contain a large number of pollen grain. Okay, and anther wall is actually composed of how many layers? 4 to 5 layers. The innermost layer of this wall develops a single layer called tapetum. Okay, and the tapetal layer is the great physiological significance, as all the food materials which are entering in the sporogenous tissue, they diffuses to this layer. Okay, so this layer, that's called tapetum layer, and ultimately the tapetal layers disorganize. After that we have studied about microsporogenesis, that is development of pollen grain. Now pollen grains, they are produced in the anther loop. This is the anther loop. Okay, that's called microsporangium. There are 2 anther lobes. Each lobe contains 2 sacs, that's microsporangium. This is microsporangium, and tapetum layer is providing nutrition to the developing material inside it. So this material is called primary sporogenous tissue, which keeps on dividing and redividing mitotically to form sporogenous tissue, and this sporogenous tissue is diploid in nature. Okay, and diploid means the division is mitotic. Now each of these cells of sporogenous tissue, they act as a microspore mother cell which undergoes meiotic division, and as per the rule, meiotic forms 4 haploid cells, so 4 haploid spores are produced. This is a pattern of division, and this is called pollen tetrad. This pattern is called pollen tetrad. We have different patterns, like they may arrange in the linear fashion also, and ultimately these 4 spores which are produced, they get separated and finally they form pollen grain. Okay,